Hello, my name is Samantha Lisi, and this is my eReport. I will be using pictures to help describe each topic being covered during this presentation. These are also referenced at the end. We are going to start with the cell membrane and its structure. The cell membrane is also called the lipid bilayer. The cell membrane is simultaneously a portal and a barrier at once. The lipids provide the barrier, while proteins provide the portals. Membrane proteins are not only pumps and channels, but they are also cellular antennas that monitor signals that are being sent to the cell and around the entire organism. They can change shape, stretch, flex, and even grow. And on that note, they can also deform without tearing. The cell membrane is self-sealing, and this means that if the membrane gets any holes or puncture, it can seal itself up without wasting any energy. The cell membrane has channels, transporters, and proteins. The cell membrane consists of channels that have 0 moving parts, and they create hydrophilic holes that can be used by only a few select molecules. Most of these channels are specifically for small inorganic molecules, and they are called ion channels. In these ion channels, molecules follow their own concentration gradient. Most channels move molecules in a downhill direction. The cell membrane also have pumps that use ATP or light to drive thermodynamically uphill transport of molecules. These pumps are an example of active transport. Transporters are proteins that consist of moving parts that can move molecules from one side of the membrane to the other. It does this by shape changing. Transporters can usually only move molecules from inside the cell to outside the cell, or outside the cell to inside the cell. Usually not both ways. And they move the molecules against their concentration gradient. The only way to pass through a transporter protein is for the molecule to perfectly fit into the binding site of the transporter. The integral membrane proteins are a permanent part of the cell membrane and can penetrate the membrane. They are associated with one or the other side of the membrane, or bacteriorhodopsin, as it is an example of light-powered proteins. As stated above, the cell membrane is also called the lipid bilayer. But why? When the lipid bilayer are in water, they have to be a finette sheet that doesn't have any free edge. The reason for this is because a finette sheet can roll itself into a self-sealing ball. The lipid bilayer must spontaneously roll itself into a ball to conserve energy. It is made of 2 opposed layers of lipids. This is what makes up the barrier between the outside of the cell and the inside of the cell. It does this by blocking diffusion of most hydrophilic molecules, so the hydrophobic tails hide on the inside of the layers facing other hydrophobic tails, and the hydrophilic heads face outwards towards the environment. Catherine-coated vesicles. Catherine-coated vesicles bud off of the Golgi body apparatus for secretion, and then bud off of the cell membrane for endocytosis. At the cell membrane, each individual vesicle starts off as a Catherine-coated pit. This pit is like a basket structure that has Catherine proteins mesh at its base. This is a process that sets the stage for the making of vesicles. A small protein called diamine will eventually form around the neck of the vesicle to pinch it off from the rest of the membrane. And once this is constricted to the point of breaking, the vesicle is free and released. This leads to how the vesicles select their appropriate cargo, but Catherine has nothing to do with this. Adaptins play a role in cargo selection. Adaptins secure the Clathrin cage to the vesicle membrane, and they choose the cargo that will be loaded into that specific vesicle. Adaptins help capture specific cargo molecules by trapping the receptors below the neck of the budding vesicles. Proteins are destined for their targets and have transport signals on them that bind to the cargo receptors. This is the vesicle membrane-bound proteins. Formation and docking. When it comes to vesicle docking, it is dependent on tethers and snares. Once the vesicle beds off, it is released from the point of origin to find its way to its destination. It will then fuse with that membrane and release the contents it contains. Once at the target, the vesicle must recognize the membrane and dock with it. When there is docking, there will be membrane fusion. There are specialized proteins that are called RABs, and they are protein cues on the surface of the vesicle. And they are what is recognized and are bound by tethering proteins present on the membrane of the destination. Each type of membrane has its own RAB protein subfamily. So basically, there is a membrane identifier. The interaction between RAB proteins and tethering proteins are needed for docking. So the system ensures that only vesicles from the correct origin are to fuse with the correct targets. Though, there are also additional ways to recognize that, and it is through the snare proteins. Once the RABs and the tethering proteins have interacting, there is an interaction between the snare proteins on both membranes to stabilize docking. Then the membranes have to be fused to connect the vesicles and the targets. This results in the vesicle membrane becoming target membrane, which means this is how membrane-bound proteins come to be. Fusion. Sometimes fusion is delayed. The vesicle docks, and it is held in place. Then it is poised and then positioned for fusion. Only when the correct signal is given for fusion, and this is true for cellular secretion. Membrane fusion requires that the vesicle and the target membrane have to come in very close contact, and this distance closer than when in docking. This occurs because of the snares twisting and winching the vesicle into place. The cytoskeleton. The cytoskeleton consists of 3 different types of protein filaments. They all have different roles. They are intermediate filaments, which are also IFs, the microtubules, MTs, and actin. We're going to start with the intermediate filaments. Intermediate filaments have super strength, and their main function is to help the cell withstand mechanical stress. They are the toughest and most durable of the 3 types of filaments. IFs are found in the cytoplasm of most cells, where they are a network that is surrounding the nucleus and extend outwards to the cell membrane. They are typically anchored on either end. When they are found in the nucleus, where they are the main component of nuclear cortex, this is called the nucleolamin. IFs are strong and rope-like. They are individual fibers twisted together into strands, and these strands are twisted together into rope. This right here is what provides their strength. The pairs of IF tend to wrap around each other and form dimers. These dimers come together with other dimers to form a staggered tetramer. And these tetramer then bundle to form the IF's rope. The protein heads are globular and can interact with many things. Since the IF strength is against mechanical stress, we find that the most abundant of IFs are where there is most mechanical stress in the body. There are 4 main classes of IFs. There's keratin filaments in epithelial cells, and these are the broadest types that are found in the cytoplasms of most cells. Keratins distribute the stress of stretching, especially in the skin and in the gut. Then there are vitamins and vitamin-related filaments. This is found in the connective tissue cells, muscle cells, and support cells in the nervous system. There are neurofilaments that provide most of the structural support of neurons. They are long, thin, fragile axons. Nuclear lamins, which involve the strengthening of the nuclear envelope of all cells. The nuclear envelope is supported by the meshwork of IFs. Lamins are extremely dynamic and must disable and reform around each round of cell division. And nuclear lamins have to disassemble and reassemble often. This process is controlled by phosphorylation. This weakens the lamin's affinity for other lamins, which causes the disassembly, and dephosphorylation causes the lamins to assemble. Microtubules. MTs play a role in organization in eukaryotic cells. They are long, hollow tubes of proteins that have the ability to rapidly disassemble at one place and reassemble somewhere else. MTs have a point of origin, unlike IFs. They grow out of small structures near the centrosomes. They extend out to the interior face of the cell membrane, which creates protein tracks throughout the cells. The walking proteins use these tracks to move the cargo around the cell. MTs are used to anchor and localize organelles. They are much more functional than they are structural. During mitosis, the cytoplasmic MTs are disassembled, but then they are reassembled at the mitotic spindles, and this brings MTs to the chromosomes and are pulled apart by sister chromatids. The subunit proteins are called tubulin, and these proteins are dimers of two globular proteins. The dimers stack together and interact through non-covalent forces to form part of the wall of the MTs. It takes about 13 of these protofilaments interacting sides to make up a single hollow microtubule. Each dimer consists of one alpha subunit and one beta subunit, and this is because this forms a protofilament, and they alternate down the filament. Each protofilament faces the same direction, giving the MTs polarity. The ends are referred to as positive and negative. The polarity of MTs are critical to their function, and if they didn't have polarity, they could not serve their function in defining a direction for intercellular transport. The centrosomes are the main organizing centers in animal cells. The microtubules are rapidly and efficiently when tubulin dimers are being added to pre-existent MT fibers. But they don't readily form tubes from scratch. Growing MT shows dynamic instability, and once it has been started at the centrosome, its positive end grows out a new alpha-beta dimers are added to it. And this happens in minutes. Then after the entire MT disintegrates, though sometimes it only partially falls apart, and sometimes it falls apart all the way to the centrosomes. Each beta dimer is capable of binding to GTP or GDP. GTP binding dimers pack efficiently into the MT fibers, and GDP binding dimers do not. If they are readily polymerized, then dimers are added to the end of the fibers faster. The GTP that are being held can split. Then this results in stable GTP caps. Whereas if it is polymerized slower, then the GTP in the tubulin dimers at the end of them is hydrolyzed before making dimers and are added. And that makes the ends fray. This tips the balance towards disassembly, and once disassembly starts, it doesn't stop, and the frayed ends become a fray fiber. Actin. Actin is found in all eukaryotic cells, and it is essential for basically all of their cellular movements. Without it, cells would not be able to crawl on the surface, perform phagocytosis, or divide in two after mitosis. Actins are more like microtubulars than they are like IFs. They are inherently unstable, but when there are other proteins bound to it, it stabilizes them. Actins are thin and flexible. Each filament is twisted into a chain of identical globular actin subunits, which all have polarity pointing in the same direction. Just like the MTs, they have a positive end and a negative end, where they can grow on either end, but growth is faster and easier on the positive end. Actin filaments are thinner, shorter, and more flexible than MTs, though. They are more abundant. They rarely exist on their own. Without associated proteins bound to actin filaments to stabilize them, they can disassemble from both ends rather easily. Each free actin monomer is bound to ATP. Once it is incorporated into the filament, the ATP is cleaved into ADP, and this reduces the strength of binding for the monomer in the filaments, which promote the depolymerization. Actin filaments must also be dynamic, so they assemble and disassemble is necessary, like the MTs. And this happens even if it costs a lot of energy. Actin filaments only persist for a few minutes after they are formed. When new actin is needed, these proteins release their monomers. Actin monomers are bound by other proteins in which their assembly and new filaments are made, though most actin-binding proteins bind to filaments that are already polymerized. Actin bundling proteins hold the actin filaments together in parallel bundles. Cell crawling via actin. Cell crawling is dependent on actin. Many cells move by crawling and not just amoebas. These cells are sensing environmental signals and then migrate towards them using actin. This uses a three-step process, which is that the cell pushes out protrusions at the leading edge. Then these protrusions adhere to extracellular surface, which the cell is crawling on, and then the rest of the cell then flows forward to these anchoring points, filling the cell at the front. These all involve actin. In the first step, it is driven by the new actin polymerization. The thin sheet like lamella pallida are pushed out, which is a dense meshwork of actin. In these actin filaments, they have their positive ends close to the plasmid membrane. There are thinner and smaller protrusions that are filopodia that have a loose bundle of approximately 15 actins, and they also have the positive end pointing outwards. Both the filopodia and the lamellipodia are extremely dynamic. The cell crawling will constantly send out structures all over the cell surface, and they are anchored on a crawlable surface. Once the lamellipodia and the filopodiola are on the crawlable surface, they need to stick, and this is reversible. Integrids allow this, as they adhere the molecules in the cell's external environment. And on the inside of the membrane, the integrids are anchored to the actin filaments directly. This means that the integrids bridge the actin to the surface being crawled on. This shows the contractile force between the actin and the motor proteins of the myosin. G proteins including CAMPs. G protein-coupled receptors, or GPCRs, are the largest family of cell surface receptors proteins, with more than 700 different types. They are sensitive to a wide range of cellular signals, and they play a role in every aspect of cell communication. Approximately 50% of drugs work through GPCRs. GPCRs are transmembrane proteins that are found on the surface of the cell. They get synthesized at the rough endoplasmic endoplasmic reticulum and threaded through the endoplasmic reticulum membrane using start and stop transfer sequences. They are then shipped to the Golgi. Then they are processed and sent to the cell through transport vesicles. This involves RABs and snares. CAMPs carry the message into the cell, and this is why it is a second messenger. CAMPs have no informational value at all, but they convey information, and they activate a single protein called protein kinase A. GPCRs with calcium as a second messenger. As for calcium, phospholipase C hydrolyzes split PIP2 into two molecules. IP3s is what serves as a second messenger, and it leaves the membrane and then travels to the cell interior. DAG remains in the membrane and then activates protein kinase C, which phosphorylates many target proteins that include the calcium channel, and this is what allows some calcium into the cell. For IP3, it goes to the ER and is triggered for the release of calcium here. The calcium then binds to the PKC, making it more active, and to the protein of calcium that becomes more active. When calcium activates another kinase that phosphorylates target proteins that go on to do other things. CDKs, or cyclin-dependent kinases, are the family of protein kinases. Cyclins are a set of proteins that control the kinases that are involved in the cell cycle. While cyclins are not enzymatic themselves, that is why they have to interact with kinases. Only when particular cyclin is made, a corresponding CDK will be active, which give the CDKs their periodically during the cell cycle. Different cyclins are made for different phases of the cell cycle. Each cyclin CDK complex triggers a different cellular event to prepare for the next stage, though phosphorylation of kinesis is what brings out the cellular response. Cyclin binds and activates CDK, and then those CDKs will go off to a target protein and phosphorylate it, which changes the cellular behavior. Cell junctions. Cell junctions are what link cells together through the act of physical contact. And there are 5 different ways to do this with epithelial cells. Tight junctions seal the cells together so that nothing leaks through, and this is formed by the claudins and the oclaudin proteins. They are arranged like a zipper. Without tight junctions, life would not be possible at all, because molecules would just leak through. Cell-to-cell mechanical have 3 that are similar to tight junctions. There are adhin junctions, desmosomes that bind one epithelial to another. Then there are hemidemosomes that bind the epithelial cells to the basal limen. All 3 basically provide a sheet of epithelial cells that join the individual cytoskeleton together with mechanical strength. Adherent junctions and desmosomes connect cells through a transmembrane protein called cadherin. Cadherin is a neighboring cell interact tightly and link 2 cells together, and this requires calcium ions. Cadherins are tethered to actin filaments on the inside of the cell, which directly links them to the cytoskeleton. While integrins and hemidemosomes are linked to collagen, there are also gap junctions that are very private and have light communication. These only exist between neighboring cells. So, rather than being sealed or anchored, they are openings that match transmembrane protein complexes that are called connexas that are met between 2 cells and create continuous channels across 2 membrane. Gap junctions are also regulated and can be shut off in response to a cue. Lastly, we are going to be talking about Cancer. Cancer is a cellular mutation. Anything that can increase the mutation rate in cells will increase a risk of cancer. Smoking is an example of something that can increase the risk of cancer. Cancer is your own cells turning against you and putting individual cells before their survival as a whole. There are 3 types of proliferated cells that create tumors. The first one being a benign tumor. This is where there's a rapid growth of mast cells, but they remain localized and usually can be fully removed by surgery. The second type of tumor is malignant, and this type of tumor has the ability to invade surrounding tissue. The third type of tumor is probably the most scary, and this is the metastatic tumor, and this is where tumors can invade neighboring tissues, but can also migrate to invade the blood. So this is a malignant cells that get into the circulatory system and start to give secondary tumors that have the properties of the primary tumors and can also rapidly divide and are extremely invasive. Since cancer is so unpredictable, there is likely a chance that we will never be able to get rid of it. But there is new technology every day that could change that. Epidemiology is a big part of cancer, and that is the statistics of the relationship between certain environmental factors of disease. Cancers are usually regional and not demographic or familial. Migrants are more often getting cancer from their new home than their old one. But cancer is not solely from the environment. What makes cancer unique as a genetic disease is that it is exclusively a disease of the somatic mutations, as they do not cause the entire organism to mutate like a germline mutation would. And if the entire organism did mutate, then this would mean that the whole organism would be made up of unregulated cell division. There are 2 classes of gene mutations that could lead to cancer, and this is oncogenes, and this is when the protein activity itself causes the disease. And then there is unmutated genes that is called proto-oncogenes. That is when they are mutated and they become inactive, and this causes cancer. This is tumor suppressor genes. In this case, usually the protein is normally protecting against cancer. When it comes to oncogene mutations and the tumor suppression gene mutations, one is dominant over the other. In this case, the oncogene mutations are genetically dominant, and the tumor suppressor genes are genetically recessive. The reason that the tumor suppressor genes are recessive is because there is genetic predisposition to cancer. And if you have 2 healthy tumor suppressor gene alleles, then that means you don't have cancer. You have to have both of these alleles mutated for you to have cancer. So that also means that if you have 1 healthy allele and 1 mutated allele, you also do not have cancer. It is important for you to know that cancer is caused by the buildup of mutations and very rarely happens from a single mutation. Genetic instability is usually the cause of a cancer progression because of the mutations go on and affect the replication and the maintenance of the genome. There are larger scale chromosomal mutations. Cancer cells are dangerous, not just because they are cancer, but because it is cancer cells that overtake the normal cells and survive better. They have a competitive edge. They will literally do anything to win. But in their case, when they win, cells die, and when too many cells die, people die. But that doesn't matter to a cancer cells. All that matters is that they overtake a person's body, and the cancer will not stop until that goal happens. Thank you, and I hope you enjoyed.